So I think we'll start the session for today. So I'll be sharing my screen. Uh, Please, someone let me know whether the slides are visible and whether you can hear my voice.
Sir, it's visible.
Okay. So yeah, so the slides should be visible now. Uh, just yeah. Okay. So for today, again, we will cover the week 5 uh, sessions content for the NPTEL course Bioinformatics Algorithms and Applications, instructed by Professor Michael Komiha from IIT Madras. And I'm Suranta Samunti, PhD scholar from IIT Indore. And we will cover some of the concepts that Professor Komiha discussed in this class. We will also do a little bit of a recap of our earlier class that we already covered. And then we will also try to solve some of the questions, right? So, okay. So in week 5's content, I think Professor Gomiha went over different, I guess, concepts related to protein sequence and structure analysis, as well as some clustering algorithms, reducing redundancy in data sets, and these types of concepts, right? Information theory related concepts as well. So we'll try to cover some of those. We'll try to see what fundamental concepts are basically there. Okay. So I guess before we begin, we will just do some of the recap for the previous week's class. So in previous week, we covered the alignment matrix as well as the weight matrices, which kind of give us the individual components of the sequence and how their position related score can be calculated, right? So this kind of position related scores, for example, can give us the prominence or occurrence of certain components in a particular position of a sequence. So just not the normal sequence alignment, something like that, right? This has position specific scoring kind of, right? So apart from the alignment matrix and weight matrices, one other kind of component was discussed. That was the position specific scoring matrix. And what the position specific scoring matrix basically does, again, it is a positional calculation of the prominence or occurrence of particular amino acids in a sequence, right? And then here again, basically following multiple sequence alignment, we have to calculate the log odds score and then to convert that to the log base of 2, right? And then ultimately, when we construct the entire matrix by including all of the frequencies, normalization, log odds score, etc., then we can get the scoring, right? And the scoring can indicate whether certain residues are a certain particular component is prevalent at particular positions, right? So that was kind of the essence of the position specific scoring matrix. Again, here, if you can see that, for example, positive scores, like for example, in this 1.15 at position 3 for G, right? So here we can see that G is quite prevalent at position 3. So that means that higher positive values can indicate that this particular component is much more prevalent at that particular position, right? Conversely, the negative values will indicate that that particular component is not prevalent at this particular position. So this can give us a position specific idea. Then we also went over concepts regarding normalization. We know that if a range of data points that we are trying to utilize in our algorithms, if that is too big, it can sometimes create computational problems, right? The computational expense will be higher. So we can a lot of times try to reduce the range of that number. For example, here between 0 and 1, we are trying to normalize certain set of data points, right? So that can be done. There are different other ways to do this. For example, converting this particular range of numbers to a log-based score so that it is, better, more manageable, and more computationally efficient to compute, so on and so forth, right? Then we also covered phylogenetic tree related concepts. So phylogenetic trees are basically your evolutionary relationships, and that can be identified by a variety of methods. For example, here we discussed the UPGMA, or the unweighted pair group method using arithmetic mean. And what kind of the general process was that first we had to, of course, we have a given set of sequences, which are also called the taxa, right? Here, taxa or taxon, right? So we have to find out, for example, the lowest pairwise distance amongst our given data set, and then we have to put basically a node there. Then we have to recalculate the kind of entire matrix, combining these two taxon that has the lowest pairwise distance as a new composite taxon. And then we just iterate through the process, which kind of gave us a tree-like structure such as this one, right? So here, for example, we had 4 taxon or 4 species. We have some sort of um, let's say sequence related information for those, and then based on that we calculated the distances, right? The distances of one particular species or one taxon from the other, and what these distances basically represent, they are kind of the difference in the sense, or you can say that they are the mismatches um, that are occurring between these set of sequences. So that kind of tells us, okay, if the distance is too close, that means that these particular species are uh, evolutionarily speaking, they are really close by, right? So they could be diverging from a common ancestor that is also staying close, right? So that kind of information can be obtained from here. For example, here the lowest was 0.35. So the 0.35 was between A and C. So in the next step, what we did is that we put a common point or a node between this A and C. The total distance of 0.35 we divided by 2. So from the common particular point, each of these species are at a 0.175 distance. And then for the next step, we combine these two taxon as a composite taxon. And here we basically did that. Now, when we have to calculate the distance from this particular composite taxon to the other ones, for example, B and D, what we do is that we calculate the individual components distance from this particular single species, and then we do an average of those, right? For example, here AB plus AC by 2, that would be 0.40 plus 0.45 by 2, and that's how we get the basically the distance from the composite taxon, right? And then again, uh, we iterate through the process, means that we again find out the lowest pairwise distance, and then we put a node between the composite taxon and this particular species, and then ultimately throughout the iteration we can get, for example, a complete tree kind of structure, right? So this can tell us, okay, these two species are close by, these two species are kind of diverging from this common ancestor point, this node is connected to that node, these types of information, right? So that was kind of the process of simple process of constructing a phylogenetic tree. Okay. So yeah, we also talked about the helical transmembrane helical segments and how to identify a stage of, for example, hydrophobic and hydrophilic and kind of uh, alternating property having regions. And then we again looked at how to represent the particular phylogenetic trees in an array-like structure. For example, in an array of, for example, notations, right? So here we learned about the Newick notation, where basically your two species, when they are connected by a common point, we have to put kind of a parenthesis between those two, right? For example, A and B is connected by this common point. So A and B are together put in a parenthesis, and then this common point is further connected to C, right, here. So then, okay, first we have taken identify the other ones. For example, D and E is put in this common point. So D and E is put here. Then D and E is connected to C. So let's put one other, one C inside another parenthesis with D and E, and then these particular common point, this particular common point, and this F, they are connected together, right? So we just again put a parenthesis while including A, B as separate point, C, D, E as separate point, and then F as a separate point, and that's how we get Newick notation, right? So Newick notation was calculated, some sort of hydrophobicity indices are calculated. We also looked at how to represent the distances from the particular common points or nodes, because sometimes it can happen. In general, in phylogenetic tree, we saw that in UPGMA, common assumption was that the evolutionary rate was staying constant. For example, the distance from one particular common point to the nodes was not considered. But here in the UPG, in this particular new iteration, we can include these sort of distance or ratio of distances as well, right? Then we looked at the rooted versus unrooted tree. So rooted tree means that a common point is indicated. Unrooted tree means that the common point is not indicated, right? And this were the formula for calculating how many types of rooted and unrooted tree topologies can be obtained if we have a certain number of species that are given. And this is kind of what it looked like, right? For example, in 3 taxa, we can have, for example, 1 unrooted tree and 3 rooted tree. so on and so forth, right? So the root entry number was kind of higher in all of these cases. Okay. And as we increase the number of topologies or the number of species, of course, the number of tree topologies will significantly increase, which means that significant computational power is required to analyze um, a large number of species, right, together in a phylogenetic analysis. So those were kind of the concepts that we went over in our earlier classes. So in this week's content, I guess before we move on for the sequence-based and structure-based kind of content, so let's try to recap what the secondary structure and the tertiary structure and so on and so forth was for the proteins, right? So, so which particular type of interaction is necessary in a way to form secondary structures? Anyone?
Sir, hydrogen bonding?
Yeah. So hydrogen bondings are necessary for the secondary structure formation, right? And we can see that different types of hydrogen bonding patterns are seen in different types of secondary structures, right? So basically hydrogen bond mediated stabilization, right? What is hydrogen bond? Hydrogen bond is basically uh, you have one electronegative atom attached to another electronegative atom, um, and then the one electronegative atom is, for example, connected to hydrogen, right? So there would be partial charge uh, separation, partial charge distribution between those two. The electronegative atom will have a delta minus, and then the hydrogen atom will have a delta plus, right? So this delta plus hydrogen atom can form this kind of hydrogen bonding interaction with another electronegative atom. So that is called a hydrogen bond, right? And then here it is written that the formation of a hydrogen bond between hydrogen atoms attached to the electronegative nitrogen atom of a peptide linkage is how the hydrogen bond is formed in a protein or peptide structures, right? And then the electronegative carbonyl atom oxygen of the fourth amino acid on the amino terminal side of the peptide bond uh, is, for example, seen in the alpha helical structures, right? So that means that I with I plus 4, that kind of hydrogen bonding pattern, if you see, so that means that you will find a alpha helical kind of secondary structure, right? Similarly, we see that in the case of beta shapes also different hydrogen bonding patterns are seen, right? So here, for example, it's saying that each successive turn of the alpha helix is held in place by 3 or 4 hydrogen bonds. And then the hydrogen bonds contribute to the stability of the overall alpha helical structure. So uh, in all of the cases, when we have, for example, tertiary fold of the protein, of course, that's the 3D fold, right? But stabilizing factor, one important stabilizing factor for certain regions of these proteins, that can be hydrogen bonding, right? uh, Hydrogen bonding can of course help forming the secondary structures, and the secondary structures will be more stable because if it is regularly in an unstructured kind of or loop kind of region, right, where there is no hydrogen bond formation, you will see that the hydrogen bond or any kind of interaction formation, you will see that it is much more flexible or fluctuating in a solvated, for example, in a physiological kind of setup. So the formation of hydrogen bonds stabilizes those kind of structures. There are other, of course, factors. For example, there could be electrostatic interactions, salt bridge formation, etc. That can stabilize the folding in certain cases, right? So, okay, so hydrogen bonds are uh, necessary for secondary structure formation or stabilization of those. So, do you remember what was necessary for the tertiary fold or the 3D fold of a protein? So, if you um, let's say considering a solvated environment, you have a stretch of amino acid sequence, and that is supposed to be folded into a 3D structure, a 3D architecture, right? So what interaction was necessary for uh, the 3D folding in a solvated environment of the protein? So I see one question, I think. Why do we skip one species from the table? Uh, see, how does that? Uh, So you see, here no species was skipped kind of. In the table, we have ABC versus BCD, so that kind of accounts for all of the species. And after we went for the tree kind of calculation here, so anyway, A, C, and B, these were combined to this BAC, right? BAC combined taxon. And then since there is only one species left, of course, species D, so the logical next iteration would be that to calculate the distance from PAC to D, right? And that we can calculate via just combining the individual distances and taking an average of that. And since there is only one taxon kind of B, oh sorry, one species left, that is D, so it is supposed, it is of course can be assumed that the D is connected to the ABC composite taxon through one common point. So we are not really skipping anything, just what is left we are including it at the end. Okay, so in this kind of iterative process, so first we calculate the lowest one, then combine that, then calculate from the combined taxon the distance to the other ones, and then that will subsequently reduce the size of the table in each iteration, right? So what is left we just include at the end. Another I see, yeah, non-covalent bonds, disulfide bonded hydrophobic interactions. Yeah, so that's correct, kind of, right? So the hydrophobic interactions or hydrophobic collapses, right? That is what kind of prompted the particular amino acids to fold into a 3D set of structure, right? So the hydrophobic interaction was driven by which kind of component? Do you remember?
Sir, hydrophobic amino acid like
Mhm.
lysine and leucine all
Lysine is not.
just clump up
Yeah.
together.
Mhm.
No,
Yeah.
sorry, sir, not lysine, leucine
Yeah.
and isoleucine.
Yeah. So they tend
So
to,
they clump up together.
yeah, they tend to clump up together in a solvent environment uh, to basically maximize or increase the entropy, right? So, for example, if you have, let's say, a chain of amino acids that you are dumping into water or any kind of polar solvent, right? Let's say these are the, this is the chain of amino acid, let's say, right? These points are representing your individual amino acid residues, right? So what will happen is that these are covalently formed kind of, so a peptide kind of structure, right? So what will happen is that since you are putting this in a polar environment, let's say water, right? So the water already had a strong hydrogen bonding network. Now, when you put this kind of uh, group of hydrophobic amino acid or a peptide, let's say in water, so what will happen is that the water will tend to surround these dots or these individual amino acids and create kind of a solvation layer surrounding those, right? Now, the solvation layer or the layer of water around these amino acids, these are basically highly ordered, very highly ordered structures. They have a good, connection of hydrogen bonds between them. So what will tend to happen is that in order to increase the disorderedness or increase the entropy of the system, the amino acids will tend to come closer and kind of tend to clump up together. Okay, kind of like this, let's say, right? Like this kind of, in a way. Because if it is kind of grouped together, so there will be a common solvation layer that will be formed around those, and the surface area or volume of this particular solvation layer will be reduced if you do the calculation. So the amount of water it takes, or the volume of water, or the surface area of water that it takes to cover individual amino acids versus one group of clumped up amino acids. If you combine the individual amino acid values together, you will find that the clumped up amino acids or grouped up amino acids together will have a lesser number of less kind of value of the surface area there, right? So that means that the ordered structure or the solvation layers surface is decreased, the volume is decreased, if you will. So that means that inadvertently the disorderedness is kind of increasing, right? So that's the logic kind of why these amino acids will tend to group together or group up together, okay, to increase entropy or to increase the disorderedness. Apart from alpha helix, of course, you have another set of secondary structures that is called the beta strand or beta sheets, right? Um, and in beta strands also, you will see that certain type of hydrogen bonding patterns are there.
Sir,
Yeah, please.
I have a question.
Mhm.
Sir, if they try to clump up together, so there will be an increase or decrease in entropy.
No, there will be increase in entropy because the surface area
Okay.
of the water layer that will be reduced. So since the surface area of water represents a ordered structure, so that means the surface area or the volume of the ordered structure is reduced, right? If that makes sense. So that means that the entropy or disorderliness
It's a,
is reduced.
I understood. I understood.
Yeah,
Yeah. So the order structure is increased, so the entropy is
no, yeah, the ordered structure
decreased.
is
Huh?
decreased.
Vice versa.
Yeah, yeah.
Yeah.
So yeah, that's kind of the logic there. So in beta sheets, for example, you will see that the adjacent polypeptide chains, they can be arranged in either a parallel or an anti-parallel orientation, right? Uh, so parallel, what it means that the chains will have the same type of amino acid to carboxyl orientations, right? For example, here you see the orientation is kind of in this particular way. If you see by this arrow, that is also representing that. Whereas in the anti-parallel orientation, you will see that they have kind of opposite orientations, right? So, for example, this strand is in this way, this strand is in this way, this strand is in this way, and so on. Now they have somewhat similar structures, of course. The repeat period for them is slightly different. For example, in parallel configuration, it is 0.5 angstrom shorter. The hydrogen bonding pattern also is a little bit different, right? So here it says that the hydrogen bonding pattern between adjacent strands differ in what way? Uh, Basically, how they are aligned together. Okay. So in the antiparallel beta sheet, interstrand hydrogen bonds are essentially in line. Now, what that means is that if you compare these sort of two antiparallel strands, right, that are forming the sheet, you will notice that uh, the hydrogen bonding, for example, if this blue one is representing, let's say, nitrogen, right? The red one is representing an oxygen. You will see that the hydrogen bond is kind of straight, right? The nitrogen that is giving the hydrogen and then the oxygen on the other strand that are forming the hydrogen bond, right? They are kind of in line, right? The hydrogen bonding is forming not at an angle, but at a kind of straight downwards pattern, right? Straight downwards line kind of way. So that is because um, due to how they are positioned in a sense. Okay, so you will see that the positioning of the anti-parallel sheet is kind of slightly skewed towards a particular direction, let's say left or right, right? So that is what allows the nitrogen and the oxygen atom to come in a straight line, whether in the anti-parallel, whereas in the anti-parallel beta sheets, we will notice that exactly the atoms that represent the strands, the similar atoms are kind of in line with one another. So what I mean by that is, for example, first of all, these are the carbon atoms, right? These gray ones. These are the side chains. So these are, let's say, the nitrogens. The red ones are oxygens. And you will notice that, for example, this nitrogen, this nitrogen, this nitrogen, they are kind of in line, right? This particular oxygen and the in the downward strands oxygen, they are also in line, right? This carbon, the side chains and etc., they are also kind of in line. So what that means is that since not the same atoms are forming hydrogen bonds, right? One nitrogen atom is and one oxygen atom sort of is interacting in this hydrogen bonding process. So that means that if the atoms are in line, so the hydrogen bonding will have to form at an angle. So that's what it means. So here, for example, this hydrogen bond is forming not straight in line, but at an angle. So this is related to the positioning of the particular amino acids or the particular atoms of the amino acids in the different strands, right? So here, that is exactly what is said. So in the parallel variant, the inter-strand hydrogen bonds may be distorted or not in line. So due to them being not in line, we will see that the hydrogen bonding is forming at an angle, and this kind of gives rise to a parallel sort of beta sheet kind of structure. Okay, so yeah, and then we also know that the dihedral angle patterns, like the phi and psi angle patterns of this particular secondary structures, they also have a little bit of difference between them. So this table particularly gives information regarding the idealized version or the idealized value of particular phi and psi angle that is tends to be seen in this sort of secondary structures. However, these are only idealized values. In reality, you will see there is a certain bit of fluctuation, right, of this kind of structures, and the range that is sort of allowed in this in a particular set of secondary structures, that is of course represented by one kind of plot. If you see in the bottom, so these are called the Ramachandran plots, right? So here in the upper picture, you will see that the different dihedral angles are represented. For example, between the N and C alpha, you see the phi angle, right? The C alpha and C, you see the psi angle, so on and so forth. And then here on the bottom left, you will see that by different shaded regions and this dotted kind of highlights, you will see that the different type of secondary structures that are possible are represented. For example, here you see antiparallel beta sheets. Here you see left-handed alpha helix, and so on and so forth, right? In when you do, for example, computational modeling of certain proteins, right? Let's say you have just predicted the 3D structure of a certain protein from sequences, right? Maybe you have used tools like homology modeling, or maybe you have used some AI-based uh, algorithmic tools like AlphaFold, right? So they have given you the coordinates of this predicted structure, and then you have uh, the XYZ values. So you can of course calculate the phi psi angles, right, between for all of the amino acids that are component of this particular protein, right? Uh, However, after that, after the prediction by these sort of models, you will have to go for checking whether the models are correct or not, right? So you have to kind of plot the phi and psi values of this particular amino acids that are part of these proteins, and then you have to see whether a large number of amino acids are in unallowed regions or disallowed regions, particularly basically not in these shaded regions of the Ramachandran plot, right? So if there are a large number of proteins or large number of amino acids that are in the disallowed regions, so what you have to do is that you have to perhaps again go for the modeling, or you have to do some sort of structural optimization. Let's say you might do energy minimization and then recheck the values whether they are corrected now, so on and so forth, right? You can even run longer scale simulations to see if the structures arising from those kind of physics based constraints in force fields, like if that is giving a corrected structure or not, right? So you have to do this kind of analysis.
Sir,
Yeah.
you said that we have to plot the coordinates of the atoms. So how do we get the coordinates of the atom by X-ray crystallography or something like that?
Yeah. So the coordinates of the atoms will allow you to calculate the particular phi values, psi values, and if you want to calculate say any other dihedral values, for example, chi1, chi2 for different amino acids. For already crystallized structures, of course, you have the X-ray crystallographic file, right? The PDB file that contain the XYZ coordinate values, position values for the atoms, right? If you do not have the X-ray crystallographic data, basically the PDB structures, right? So what you have to do is you have to go for modeling. Now, how do these modelings work basically? So there is one process that is called the homology modeling. So this is let's say related to homology, right? Basically, what it means that let's say you have one particular amino acid sequence which already has predicted structures, predicted structure in the PDB database, right? Now you have another particular amino acid sequence for which you do not have a known structure. So no real data is there, no experimental validation or experimental data is there for this particular amino acid sequence. If there is a very close homology, or let's say high number of let's say sequence similarity value between the sequence one and the unknown sequence right now. So basically, what you can do is you can uh, predict the structural information, or particularly the 3D coordinates of the atoms from that homologous protein which has the structure already available. So that is called homology modeling. Uh, there are other ways. For example, you can use something called like AlphaFold, like basically AI-based tools, which have been trained in a way that they are able to predict the structure from the unknown sequences, right? So that will also give you the 3D coordinate values, the X, Y, Z values, which ultimately we can use to calculate the dihedral angles. Okay.
Okay, sir.
Yeah. So the X, Y, Z coordinate values for each atom, that is kind of necessary in this case to get the values. That's why if there is already available PDB structure, that's fine. But usually the AI predicted models are not always very good. Sometimes, right? They have their own metrics, so you have to always check that whether what kind of confidence level I am getting for this particular protein or different regions of this protein. Um, Even if you are getting, let's say, low confidence level or low confidence code for certain part of the protein, you should go for structural optimization and these sort of approaches. First, you check through Ramachandran plot that whether they are having good favorability or not. And then you further go for structural optimization to see if you are getting better structures, better corrected structures by, let's say, performing energy minimization and such things. Okay. So that's one thing. Um, okay, so let's do one question quickly. So which of the following could be not true for secondary structure, secondary protein structure? So whether hydrophobic character of amino acids are important to forming the secondary structure, whether intermolecular hydrogen bonds are important in forming secondary structures, and whether these are the given secondary structure examples or all of the above. So remember, which of the following is not true? It is asked here. Okay. So always be careful about such questions. The question that is the statement that is given in the question, that is itself sort of negative, right? So which of the following are not true? So look at it in just a bit. Okay. Yeah. So yeah, a lot of you are saying D. Uh, So basically, yeah, the answer here, as stated, was not true about secondary protein structure. Now, the answer A, what it is saying, the statement A, it is saying that the hydrophilic or hydrophobic character of amino acid residue is important to forming the secondary structure. Now, remember what we earlier just discussed is that for forming the secondary structure, the important thing was the hydrogen bonding pattern, right? The hydrophilic and hydrophobic characteristics, they kind of come while the formation of the tertiary structures or the 3D fold of the protein. Okay, so that's where the entropy related concepts are there, and the hydrophobic parts will kind of tend to stay away from the solvent exposed area, whether the polar groups are kind of staying to the surface, right? They want to be solvent exposed. So that is coming in the 3D structure formation segment. However, what the statement B and C are saying is that, so the B statement is saying that the amino acids, ability of amino acids to form intramolecular hydrogen bonds is important in secondary structure, right? So that is a true statement, right? Because the intramolecular hydrogen bonding pattern is the determinant of the secondary structure, one of the determinants of the secondary structure, right? And then the statement C is also saying that the alpha helix, beta platelet sheet, beta trans are all secondary structure parts. So that's also correct. So in this case, I think option A should have been the best answer, because the hydrophilic hydrophobic part or characteristics, they come in formation of the 3D fold, not so much in the secondary structure formation. Okay. So be careful about this kind of questions, this kind of slightly analytical questions. So they might also come in your assignments. Okay. So let's say this kind of question is also quite regular. So it is asking that which one can best describe the reason for association or clustering of hydrophobic residues in an aqueous solution. So we just discussed, I guess, a little while ago. So what is the answer here? Yeah, so option C, right? A favorable increase in entropy. as we discussed earlier, due to the clumping of the hydrophobic amino acids or groups, there will be an increase in entropy. Okay, so that is the reason for clustering of hydrophobic residues in an aqueous solution. Okay, that's fine. So one concept that Professor Bromiha discussed in this set of classes was non-redundancy, right? So what is it meant by non-redundant data sets or data points? Anyone? So what does non-redundancy indicate, and is that important or not? Overlapping or recurring data? Yeah, basically, kind of same. Recurring data, right? You are saying. Uh, so a redundant data set will mean that similar type of data points, or perhaps the same data points, are uh, included multiple times in a particular data set. So what that could do is that could kind of skew the inference that we are wanting to make from this particular data set. Okay. So how can the data set, or how can the outcome that is taken from the data set be skewed by including non-redundancy? Let's say we have this particular example that Professor Rumiha also gave in his lectures. So let's say we have two sequences, right? We have let's say this is seq1, and then we have seq2, right? So what we might want to calculate in a given particular data set is that we can calculate the um, occurrence or the composition of particular amino acid through an entirety of our data set, right? Let's say you are trying to figure out from a combined um, data set of, let's say, some organisms, and you have the sequence data available there for multiple different proteins, and you are trying to see that, okay, whether hydrophobic amino acids or hydrophilic amino acids or one particular type of amino acid is more prominent than the other, something like that, right? So for example, in a very simple case, let's say we just have these two particular sequences in our data set, and we are only interested, okay, so what would be the composition of particular amino acids in this particular data set that we have, right? So let's say we try to calculate for this amino acid alanine. Okay. So can somebody tell me what is the composition of alanine in this particular data set? Composition of alanine means that you have to count how many alanines are there, and then you have to take the total combined number of amino acid that we have in this data set, and then we have to calculate the composition, right? So can anybody tell what would be the composition of alanine in this particular data set where we only have two sequences? Okay. So just how many alanines are there divided by the total number of amino acids present. Okay. Uh, so I see 0.3 in seq1. So don't calculate for just sequence 1, sequence 2. Just take all of them together, okay? Because here we are trying to calculate for the entire data set what is the composition of alanine, okay? So not just specifically sequence 1, sequence 2, but combining that, what would be the value? Yeah, so 0.2 I see. So basically, how many alanines are there? For example, here we have one alanine, here we have another, here we have another. So 3 alanine in sequence 1, and then in sequence 2, we have 1 alanine, right? So total number of alanine is 4. Total number of amino acids, we have 10 in each, I guess, right? So total would be 20. So composition of A in this case is 0.20. Okay, that's fine. Now let's see, in this particular data set, we are including one additional sequence which will be of very high similarity with one of the components, or one of the data sets, one of the data points that we have, right? Either sequence 1 or sequence 2. So let's say now here we have included the third sequence. So in this case, we had this one, right? Seq1 as our original one. We had this one, which was Seq2. And then now we also have Seq3, which is the newly included sequence in this particular data set, right? We have just added another one. But if you notice carefully, you will see that the sequence 1 and sequence 3, they have very high similarities. For example, AEAI, that is the initial 4 characters or initial 4 amino acids of the sequence, is matching with sequence 1, in between sequence 1 and sequence 3, right? Both have AEAI. And then again, we have D in the middle at the same position. Then we have GA at the end for each of the sequence in sequence 1 and sequence 3, right? So now, since we have added a similar sequence to the data set, what would be the new composition of alanine, right, in this particular case? So can you calculate quickly? What would be the composition of alanine now that we have added a new sequence, sequence 3 in our data set? So try to do it. We'll just look at it. So yeah, we see 23%. Yeah, so you just count out total number of alanines, right? A. So we have 3 in sequence 1, and I guess A, we have 3 again in sequence 3. We have 2 in sequence, 1 in sequence 2, right? So the total number is 7. So 7 divided by 30, that will be 0.23. Okay. So what we see in this case is that by adding another sequence, which was already very similar to one of our earlier sequence we had, the total number of the total composition value of alanine is changing, right? It's shifting. Uh, it became 0.23 from 0.20, right? So let's say instead of something very similar to sequence 1, we now, let's say, do a little bit of experimentation with adding something very similar to sequence 2. So in this particular case, in the third example, we had seq1, right? That was the original one. We had seq2, that was the original one. Let's say in this case, we are adding a sequence 3, which is now again very similar to what we had in seq2, right? So the sequence 3 is very similar to sequence 2. So now what would be the composition of alanine? Can you try to do it? So we see 0.16%. So how many alanines are there? It is 1, 2, 3, and then in this case 1, in this case 1, so total 5, right? So 5 divided by 30 is 0.17, 16.7, that's very close to 17%, right? Uh, so yeah, so again here we see that the from the original composition 0.20, the composition is decreased now, right? And that's because now we have a new sequence which does not have that high prominence in alanine, right? Now that's fine, but since here sequence 2 and sequence 3 are very similar, to basically make the data set better, or in sometimes not very biased, right? If we have, let's say, large number of sequences in our data set, what we can often do is we can include some sort of threshold value. So we can say that, okay, so if there is a threshold value, and if two sequences are, for example, exceeding in similarity this particular value, a threshold cutoff, then we will only consider one of those sequences to be included in our data set, right? For example, let's say if we have a data set of kind of 1,000 amino acids. So let's say there are like 60 amino acids that are having greater than 90% similarity, right? Or identity, let's say. So that means that the inference that we can draw just based on these 60 amino acid sequences, that can be skewed by a lot, right? So we can, let's say, take some sort of threshold value, and we can say that, okay, so if it exceeds, let's say, 90% identity value, 90% similarity value, then what we can do is that we can only include one of those amino acid sequences as a representative of that particular group, right? So this kind of way of grouping is called clustering. Okay, so we can cluster the sequences based on their content, amino acid composition, uh, and we can, if for example multiple sequences are coming under the same cluster, that means that they have a high sequence similarity value. Okay, so in this kind of way, we can increase or reduce the biasness of the data sometimes. So it's very important to know. So it reduces bias. Okay. One particular software or program that Professor Brumiha mentioned for reducing this kind of data sets, can anybody remember what was the name of the software or the program?
CD hit.
Yes, CD hit, right? So CD hit was mentioned by Professor Gomiha, and CD hit is basically it's called cluster database at high identity with tolerance, right? Now, what is the process of CD hit? So CD hit is basically first it's sorting the sequences that you are giving by decreasing length. Okay, so that's number one. And then what it is doing is that the longest sequence that's becoming the representative of the first cluster. Okay, so now after ordering the sequence in terms of the length, so the longest sequence it is taking, so that's your cluster one. So that sequence is cluster one. Now all of the other sequences that are now compared with this particular cluster one representative, and then it says that okay, based on the threshold values provided, whether this particular amino acid sequence, the new one, that is significantly different from the cluster one representative, or it should be a part of the same cluster or cluster one, right? So that it takes, if it is significant, significantly different, significantly high dissimilarity, or let's say relatively low similarity, so then that means that sequence will form a new cluster. Otherwise, that sequence will be joined as part of the same cluster. Okay. So again, it's sort of taking each sequence at a time, let's say, and then it is putting in a particular cluster, or it is seeing whether that sequence can form a new cluster, right? So basically, how the algorithm works is that it uses something called a short word filter. Okay, so that means that it utilizes word counting to skip pairwise alignments below a certain similarity threshold. Now, how do you find out that if two sequences are similar or dissimilar, right? You have to first align the sequences, right? You have to perform sequence alignment, right? If you have a very large data set where a lot of data points are there, let's say thousands and lakhs of data points are there, right? It could be. So if you want to align the sequences, each sequence with one another, right? So it will take a large uh, time, of course, and a large sort of computational resource, right? So the time and the computational expense will be very high. So to kind of mitigate that, CDHit uses something called the short word filter. Now, what is short word filter? So let's say we have just two representative sequence. So let's say I'm just writing it down, right? Um, okay. So let's say we have these two sequences, right? This is seq one, this is seq two. You will notice that if two sequences are very similar, for example, these two sequences are very similar, right? You can see that in similar positions they have same amino acids for majority of the positions in these sequences, right? So if two sequences are very similar, you will see that not only they align, of course, overall very well, but you will see apart from the overall alignment, you will see that there are short stretches of subsequence, or short, let's say you can say stretch of characters that we will find also that are matching here. For example, in this case, you will see that of course AGT is matching, right? So in this case, you can see that, for example, MASL is matching, right? So short stretch of subsequence or short stretch of words will also be matched. Short stretch of characters, let's say, will also match between the sequence one and sequence two. So what that means is that rather than depending on the alignment, the algorithm can find, okay, if this particular uh, three word or three character word is uh, kind of matching between sequence one and sequence two, and we see multiple number of such kind of matches. So that means that this sequence one and sequence two could be very similar, and that could be part of a same cluster, right? So this type of logic the algorithm is using, and then based on a certain kind of threshold, for example, pentapeptide for approximately 70% similarity threshold, the algorithm will operate. So you have to provide the threshold value as well as you have to provide how many character words you want to match Um, and basically, uh, some statistics is there, is that, for example, if you have, um, let's say, 80% identity between two sequences, right? You will find that certain number of words are always matching. Okay. Uh, if you have, let's say, 70% identity between two sequences, you will find again that this a certain number of this particular character match you will find in between those sequences, right? So based on that, the algorithm is kind of operating, and there are certain limitations here as well. For example, here it is saying that below a specific threshold, you cannot apply a short word filter. And what are those thresholds? For example, 80% for pentapeptide, 75% for tetrapeptide, 66% for tripeptide, so on and so forth. Now, why you can't apply uh, the particular word filter below a certain threshold is that, so let's say, the examples that are given here. So you are trying to match a certain number of characters as words between two sequences, right? So in the worst case scenario, what will happen is that, of course, if the uh, characters or amino acids are matching at different parts, so that means that there will be certain mismatches also, right? Because it's not completely matching, of course. In the worst case scenario, the mismatches can be spread very evenly across these two sequences. So what that will mean is that uh, you cannot find a proper match using a certain stretch of characters, if that is the case, where the mismatches are distributed evenly. For example, the best, I guess the easiest one to understand will be, for example, option D here. Okay, so in this particular case, you are seeing that the protein A and protein B have 50% identity. If you can see here, for example, the M versus M is matching, then V versus I is mismatching, then the G versus G is matching, D versus E is mismatching, so on and so forth, right? So what that means is that you will see one mismatch is appearing alternative to one match, right? One match, one mismatch, another match, another mismatch, so on and so forth. Now, in this particular sequences, you cannot find a two-word match if that is the case. Okay. Uh, do you see the logic here? So if protein A, protein B have 50% identity, and the mismatches are distributed evenly, we will never find a two-word match in this particular case. Because, of course, one mismatch is appearing alternative to one match, right? That means that no consecutive two words are matching at the same time, in a particular position. Okay, so you cannot find that. Similarly, for example, if you see that protein A and protein B are matching with 75% identity, let's say, so that means that the mismatches, if they're distributed evenly, you see that you cannot find a four character match between protein A and protein B. Of course, here you can see that the three character match is present there. But whenever you try to calculate or go for a four character match, you will see that you always find a mismatch. So it is not possible to match those, right? So these kind of limitations are there. So for example, 75% match between protein A and protein B, you have to use a three-word filter in this case. Okay. So that's the kind of logic here. So based on that theoretical limitations, you always have to kind of operate within that limit. Okay. So here are some sources that I have also given. So these are basically the sources to the CD hit sort of website and their GitHub page, I guess. So there you will find the instructions for installations and how to use those. So that is the paper. I think I have given a link to that also. So this was the original paper. This is the CD hit programs page, of course. You can go to the download section. Again, that is referring you to their GitHub pages, right? So this is what the GitHub page looks like. You have a lot of instructions here for installation, for running the program, so on and so forth. And then if you have Conda, so Conda you can get from anaconda.org. You can also install the CD hit program using Conda, right? For example, here it is saying by Conda CD hit. So that's one thing. Uh,
Sir, can you provide the link of the CD hit page as I was not able to find it online?
okay,
I
so
don't know why, but
I guess I'll just copy and paste it in the chat. And let's say copy and paste the link of the paper on the chat also. So in the paper, you will find all of the links, relevant links that is. present. Okay, that is
Okay, sir.
for this particular program. I think it is the exact one. Yeah, yeah. So let's try to quickly see one example. Okay, I guess I'll just stop sharing from this particular screen, and then I'll share. Another screen on another system. And okay, anyway, yeah, I think now you can see this particular tab or terminal here. Okay, so we have, let's say, installed the CD program. We have followed the instructions given in their web page or their particular GitHub page, right? And then we now want to use the program. Okay, so you will ultimately get like a zip file kind of file like here. So you see this .tar.gz. Uh, when extracting this file, you will get uh, a folder where a lot of different tools or toolkits that are available within the software that is given as separate files, right? So you have kind of in this particular Linux system, you have to go and run, let's say, make command for within this particular directory or folder, and then you will just ultimately get you will be able to use this program. Okay, so that's the program, but let's say what we can give as input for this particular software. Okay, so in this case I have already uh, curated, let's say, one input.dat file. So what is it is basically uh, sequences, right? It is sequences for the different kinases. um, as you can see, for species 1, it is the human genus kinase, right? The species 2 is CDK2 kinase of human cyclin dependent protein kinase 2. CDK3 is again an isoform of cyclin dependent kinase 2, sequence 3. And the sequence 4, sequence 5, you can see different kind of kinases are there. For example, MAP kinase, homodomain interacting kinase, so on and so forth. I believe a total number of 19 sequence, around something like that, should be here. Okay, so that is our input sequences, and we just want to give it to the program, and we want to say that, okay, cluster these based on their sequence similarity. Okay, so what kind of so I already had run this. Let me just delete this files, okay, because those will be generated upward after running the program. So here we have this command. Okay, so if you have already, so for this Linux system, if you have included cd hit in your bash RC, you can directly use a command called cd hit. If you have not done that, so there is this particular folder. If you see earlier, so within this folder there is one executable file called cd hit. Okay, so you have to run that particular file in this case. So what we can do is we can give the folder's path, and then we can give the particular cd hit programs command, and then we have to mention the parameters. So firstly, using -i, we can give the input.dat, the input file that you want to feed the program. Then we have to mention one output sort of file name. So -o, let's say we give output.dat, right? Then we have to mention particular parameters. For example, -c 0.9, then -n5, then minus capital M we have 8,000, then we have minus capital T 4. So the minus M 8,000, this particularly means that it is kind of a limitation on how much memory the program will be using. That's, let's say, 8,000 MB, right? 8 gigabytes of memory. And then this minus T is how many CPU cores we want uh, for this program to be used. Okay, so that is minus T4, so 4 CPU cores it will use. And then these two particular parameters, minus C.9 and minus 9.5, are what was relevant to our discussion earlier. So anybody knows what this minus C 0.9 and minus N 5 indicate? So remember that in discussion, we just mentioned that there are two parameters that we kind of have to provide. One of them was sequence identity, right? So what is the cutoff value or threshold value for that that you want to consider? Another one was the basically your word count, or how long stretch of characters you want the algorithm to use as the word filter. Okay, so in this case we are saying that okay, it should have a 90% identity threshold, and the 5-word filter process should be used for clustering in this particular case. So if we run this particular program here, we will see that since we only had a small number of sequences, so it ran very quickly. There are different informations regarding the calculations given here. For example, CPU time was very, very low, right? It also says the longest as well as the shortest sequence length, then total number of letters that was calculated, that it also mentions. Total number of sequences 19, as I said earlier. Uh, you will notice that after running the command, there are two different types of files given. For example, one will be this output.dat, output.dat file, right? If you go to the output.dat file, if you count the number of sequences, you'll notice that now, if you compare with our input.dat, the number of sequences have decreased. So where did the sequence go? That is because certain sequences are becoming part of the same cluster. So if you open the other file, the output.dat.clstr, you will see that it has a total number of 17 clusters that was identified. Okay. So of course, the numbering is from cluster 0 up to cluster 16. And you will see that in cluster 6, 3 different amino acid sequences are grouped together. Okay. So these are the KSYK, or the splinteration kinase of human, mouse, and then I guess rat. So the 3 different species, same protein. So of course that will have sequence identity of a very high level, right? That means that it is, of course, logically speaking, that should be the case, that they are kind of grouped together as part of the same cluster. So that is what the algorithm has done. And then the other particular sequences that has been provided here, the algorithm didn't find enough match or enough identity between those for them to be grouped as part of, let's say, similar clusters. Okay. So that's why there are a total of 17 clusters here. That's how this algorithm is kind of functioning. So you can use again if you are in need of some sequence similarity calculations. So of course you can use this program. CD.
Sir, I'm not from a computer background, but I have downloaded the CD hit as a zip file.
Yeah.
But that was opening in the Word section, so in my computer.
So you are using a Windows computer or Linux computer?
No, I have Microsoft.
Windows. So I think there should be some instructions. So if you have the zip file, there I think should be some instructions uh, in their download page here, or in their GitHub page of how to install it in particularly
Okay,
in
sir.
Windows. So another thing I guess you can do is that you can just download Anaconda from Conda from this anaconda.org, right? Anaconda, Miniconda, something like
Okay.
that. And then from that you can try to install the software. Okay, that is also one process, I guess.
Okay, sir.
Yeah, maybe that will be of help. So in my case, we had the Linux system, so it was kind of an easy installation.
Yes, sir.
Yeah, for Windows, it should also be kind of the
I
same.
don't have Linux in my computer.
Okay, so I'll have to check here. That's the option preferences. Okay, so again, I have to check, and then I guess I'll let you know whether it is available for Windows or not. Okay.
Okay, sir.
Yeah, yeah. Okay, just I have to check it. Okay, but that was kind of the basic. Uh, yeah, share the screen from the other system here, and yeah. So hopefully the slides are now visible again. Yeah, so CD hit basically uses a short word filter, and then there are also some issues with the algorithm that it uses. For example, if y is more similar to b but first hits a during the clustering, that y could end up in cluster number one instead of cluster number two, where b is present, right? So there are also ways to mitigate this sort of limitations. But yeah, so another okay, so this is one question regarding city hit. Another program was I guess Professor Promiha included was that is discussed later. Okay, last class, right? That's another program, but we'll look into it a bit later. So in this particular question, it is asking what does minus c and minus n indicates in this particular cd hit command. So we just talked about a different variation of this type of command, right? So what does minus c indicate and what does minus 4 indicate in this case? Minus n4 indicate. So minus c of 0.9, that means, yeah, I see one answer, threshold, yes. So minus c of 0.9 means that 90% identity for the clustering threshold. Okay, and minus n4 is the word count, exactly, yeah. So n is the word size of 4. The word filter that the program is using is word size of 4. Okay, that's fine. So one other clustering algorithm that Professor Promiha mentioned was k-means clustering. Okay, so what is k-means clustering? Now it is again another way of grouping different data points together in multiple clusters, right? So if you, for example, in this case, if you look at the distribution of the data in this particular 1D sort of distribution diagram, let's say, so you will see that of course there are 12 data points present, right? Now, visually speaking, how many groups of data points you see in this particular case? So what we have present here, let's say these are some sort of values across a kind of 1D, uh, at a 1D level, let's say, we only have one value to consider, right? We have 12 total values. Just visually speaking, what do you think the number of clusters should be? Yeah, 3, right? Because, you know, these 4 seems to be grouped up together, right? These 4 data points should be grouped up together, these 4 together, and these 4 together. So just visually speaking, there should be 3 different clusters, but that's a crude way of analyzing the data, right? So we need to somehow find out algorithmically or via calculation whether that is the correct way of separating these data points into these particular clusters, right? So how does k-means clustering do that? So of course, by observation, we see 3 clusters, but k-means clustering, what it does is that it groups the clusters based on uh, the representative cluster center and the distance of each data point from that representative cluster center, let's say. Okay, so how to do that? For example, so in this case, step one would be uh, we have to first choose the number of clusters that we aim to identify in our data. So let's say we want to have 3 clusters. So here we have k equals to 3. So how do we identify the number of clusters? We'll also see in this in an upcoming slide. But let's say we have already selected that, okay, 3 clusters should be there in that's defining our data set, right? Now, what the algorithm will do is that it will randomly select 3 different data points. Okay, so let's say here we have selected randomly 3 different data points. One is this blue kind of data point, one is green, one is red. So these will be the representative of your cluster in the first, let's say, iteration of the calculation. Okay, now from these randomly selected data points, the k-means clustering will, what it will do is that for each other data point that is present in this particular data set, it will allocate each of those data points to be part of either cluster 1, the blue cluster, cluster 2, the green cluster, or cluster 3, the red cluster. So basically, it will allocate all of the data points, the remaining ones, into a particular cluster. Okay. Now, how it will allocate that? Again, as I said, based on distances, right? So, for example, let's say this particular green dot, right? So this is the very first data point that we have. So in which category do you think this green dot, this particular gray dot should belong to? The blue cluster, the green cluster, or the red cluster? Based on the distance, the blue one, right? Because it is the closest uh, to the blue cluster, right? Blue cluster representative. If you see, the distance between this gray one and this blue one is very short, whereas the distance between this blue one and this green one is large, and the distance between this gray one and this red one is even larger, right? So the gray cluster or the gray data point will be part of the blue cluster. Okay, that's fine. So the distance is very short here, medium here, and then very large here. Okay, that's fine. So now one data point is allocated. You have to do that for each other data point available in this data set, and that is how we will get let's say allocation of each single data point into a part of some cluster, right? One of the clusters that we have indicated. Now what the algorithm will do is that it will compute the average or mean value of the data points within each cluster. So what that means is that uh, what it will do is that, let's say the data, the cluster blue, it has two data points. So what will be the average point or average kind of position or average value of these two data points that are part of the blue cluster? So that will be indicated by this sort of horizontal line, right? Or the vertical line, the pipe kind of, right? So that is representing the average or mean point of these two blue data points that are part of this cluster. Okay, so this one is indicating the average value of the green cluster, and then this one is indicating the average value of the red cluster, right? So in the next step, uh, what K-means clustering will do is that it will again iterate through the process, but now it will consider this common midpoint as the representative of the new cluster centers. So it will again allocate all of the available data points. Now again, starting from fresh, from the newly appointed cluster centers, and then it will try to see, okay, what kind of distribution we are getting, right? For example, again, this particular gray one will be part of course, cluster blue. This particular will be part of cluster green, of cluster green, right? So it will do the allocation process again, and then it will see what kind of distribution we are getting. Okay, so now here you see that a new iteration of the calculation was finished, like a new set of calculation was finished. However, you see that there was no change in how the data points were allocated in the different clusters if you compare it with the previous calculations outcome, right? So in the case of the previous calculations outcome, you will see something like this was there. In the case of the recent calculation, you see the same result was obtained. So that means that now what the K-means clustering algorithm will see that, okay, so this iteration is finished. We are not getting any new way of rearranging the data or regrouping the data from this particular iteration. So that this particular iteration is completed here. Okay. Now, when a set of iteration is completed, K-means clustering has to quantify the variance within this particular sort of allocation. So how it will do that? It will evaluate it will basically evaluate its output by summing up the variation within each cluster, and it will calculate a total variation within the cluster. So the total variation, let's say, looks like this. So this is kind of the latest appointed center of the clusters, and these are the representative data points from this particular cluster. Let's say this is the deviation range overall, right? So that means that all of the deviation ranges are added up together to get the total variation within the clusters. Okay. Now that is done with one iteration, what the algorithm will next do is that it will again repeat the entire process, starting from selecting the random cluster representative from the data set, then again allocating each data points, then again computing the mean of the available clusters, and then it will again do the process again to see whether there is change from the previous calculation outcome, so on and so forth. And then when another iteration is finished, what the k-means clustering algorithm will do, that again it will calculate the this particular iteration's total variation. And now what it will do is that it will compare with the previous iteration's total variation with the current iteration's total variation. And if you see that if there is a decrease in the current iteration's total variation, so that means that the algorithm is the new clustering that was done in the second iteration, that was better than the previous iteration. Okay, so that means that we are getting better result in each new iteration. But after a certain time point, uh, there will be one particular iteration you will observe where there is no new change occurring in each new iterations. Okay, so that means that the algorithm will be considered as converged, because subsequent iteration calculations will not give you a better result. You are not able to reduce the total number of variation within the dataset by performing new calculations. So that means the algorithm is converged. Okay, so you can see these two different links that I have given also for this particular kind of calculation. Let's say we can take a quick look at this bottom one. Uh, just
So total variation is calculated via the calculating mean.
Yeah. So
Okay.
in this particular simple process, it is just calculating the distance, um, let's say the total distance of all of the data points from the cluster representative. Okay, and then it is summing up that. So you can think of it in that way, because ultimately in this case you will see that, for example, um, let's say in this case from this particular cluster representative, if you calculate the distance from the cluster center from this data point to the cluster representative, you will get this distance, right? Then from this particular data point, you will see you will get this distance from the cluster representative, so on and so forth. So you can think of it in that way also, or you can think of it, for example, in the case of the larger red cluster, you can see the total kind of breadth of distribution that is present within the data. So that way you can also see how the variation is calculated. Okay. So the main point here is that the algorithm tries to reduce the variation at each subsequent iteration. If it is unable to do that in each new iteration, then that means the algorithm is converged. So that will give you the final result. Okay. So if you just, for example, copy paste it here, this particular link. So here it is a blog post about visualizing k-means clustering, right? So let's say we have to pick the initial centroid. So let's say I'll choose. Okay. Let's say it's a Gaussian mixture kind of thing. So let's say here we have three different groups of data. Let's say we'll just try to give the initial cluster representative somewhere here, right? Let's say this is first we are defining the red center, then the blue center, then the green center. Let's say we are doing it something like in this particular way. So now if you press go here, what it will do is that it will you will see that at each calculations end, it will adjust these particular data points or this particular cluster centroids, let's say, and it will see that they will kind of move closer to this particular clump of data that is present there. Okay, so that means that it is adjusting the position of the new centroid based on each subsequent calculation by reducing the variations within the data. So if you press go, you will see that first it will allocate all of the uh, dots or the data points as part of the different clusters, whether green, blue, or red. Then it will update the position of the centroid. Of course, you will see that now the centroids are moved a lot closer, right? Then again, it will reassign the data points as part of these clusters in the new calculation. Then update centroid, reassign points. Here it is not changing because it has already found the optimum way of grouping this particular data set. But if it was something different, then you would have seen that it was kind of uh, changing at each subsequent positions. Let's say we start randomly with um, something like an uniform points here. Okay. So let's say add centroid here, add centroid, add centroid. So let's say this kind of three centroids are given here, and let's say we just press go for visualizing. So first it allocates all of the data points as part of the particular cluster, red, blue, or green. Then it will update the centroid. You will see that the centroid and its coverage has been updated in this case. It will again reassign the data points. If you saw in the earlier case that there were some blue dots present here, but now it will consider that as part of the red cluster. Then it will again update the centroids And now you will see in the coverage of this particular date cluster, these blue data points are also coming, right? So if you reassign the points again, those blue data points will be also be part of the red cluster right now. So it will again perform the calculation in this different way. And after a certain point, you will see that no new kind of allocation you are getting. So for example, right now that seems to be the point where in each new steps, you are not getting any new variation, right? So that means that the algorithm has converged, and this is the kind of optimum division of this particular data set into the clusters. Okay. So that's kind of the k-means clustering process. So one question will come is that how do you know how many number of clusters you need to basically divide your data into, right? So that is one process. There are other processes also, but one commonly used method is called the elbow method, using something called the within cluster sum of squares. Now, what is the within cluster sum of squares? Well, it can be represented by this formula. So here, basically, what you have to do is that you have to calculate again the distance from the cluster centroid of a particular cluster for each data point, and then you have to kind of square that up, and then you have to do a summation for that those squared up values, right? And you have to do that for each cluster that is present after dividing your data set. Okay, so if you do the calculation for a number of clusters, um, let's say number of total clusters. For example, here you are seeing that, okay, if we are dividing our data set into one cluster, if we are dividing into two cluster, three cluster, four cluster, five cluster, and so on and so forth. So what will be the within cluster sum of square value for all of those cases? And then what you can do is that you can plot this particular data points of the number of cluster versus the WCSS value, and it will kind of come at a point where after a certain number of clusters, you will see that if you add another, let's say one more cluster, you are not getting a significant reduction of the WCSS score. Okay. So in this particular case, in this bottom left figure, if you see, of course, cluster one had a very high value of WCSS score in this particular position. Cluster two, again, there is a significant reduction of the WCSS score. Cluster 3, again, there is also significant reduction of the WCSS score. But onward from that, for cluster 4, cluster 5, cluster 6, you are increasing the number of clusters. You are not getting the much reduced WCSS from the previous number of cluster value, right? So that means that in this particular point, where basically your WCSS plot is flattened out, you are not getting more variation if you include a higher number of clusters. So that particular point is called the elbow point. Okay, so that is why this is called the elbow method. So in this particular case, the optimum number of cluster will be 3. So for grouping this data, 3 number of clusters should be used for the calculations. Okay. Um, the above plots are basically WCSS value, and why basically you have to particularly choose via this method and number of clusters, why you can just consider uh, let's say all of the data points as part of different clusters, because that will ultimately give you the best WCSS score, right? Because not the best, but that will allow you to kind of see all of the data points individually. But in this case, if you have a minimum WCSS score of 0, for example, yeah. So in this particular case, in the left-hand side plot, so I said that mistakenly. So in this case, each separate data point is considered as its own cluster. So of course, that means that for each of the representative data points, because it is the same as the cluster centroid, that means that your distance that you're calculating there, that value will be 0, right? If you sum all of if you sum all of them up, it will still be 0. So that means that your entire WCSS value will be 0. If the goal of the elbow method is to, or if the goal of the of calculating the number of clusters, or in a k-means clustering algorithm is to reduce the variation, that means that a 0 WCSS score would be preferable, right? But in this particular case, you will see you cannot infer, even though you are getting the best WCSS value, if you are getting basically the lowest possible WCSS value of 0, you cannot infer anything from this particular set of clustering, because you have a total number of how many thousand clusters, right? A huge number of clusters is there. You cannot infer anything significantly. Similarly, if you have only one cluster, your WCSS score will be maxed out. Again, you cannot infer anything significantly from here. So that means that you have to select a particular number of clusters where not only you get significant number of variance, that is significant amount of variance that is captured, but it is also significantly kind of small enough where you are able to infer something basically from this particular set of clustering. Okay. So I'll just give you one example again. Um, so again, I will just try to log in with another screen or try to share my uh, data from another screen. Okay. So we will look at the k-means clustering process. Okay. Now, what we have in this case is that there is a particular data set that we have. Okay. So you don't need to learn about this particular data point. So this is basically this particular data set includes um, a sort of dimension reduction method from the molecular dynamics trajectory data. So you'll see that corresponding to each column, which is considered as one particular frame or one snapshot from the molecular dynamics trajectory, we have four different values. Okay. Column 2, this one, mode 1, it's stated. Column 3, it's stated as mode 2. Column 3, it's stated as mode 3. Column 4, it's stated as mode 4. Okay. So let's say we are only interested in this first two columns. So that will give us a 2D representation, right? In the example, we considered, we looked at basically a 1D representation of data. So 1D array kind of, right? So there is only one data point to be considered, one type of value to be considered. Here we will consider two different types of values that are represented in these two different columns, right? The second and the third column. The first column is including, the first column is indicating the frames from the trajectory. You will see that in this analysis, we had total of 80,000 snapshots. So corresponding to each snapshot, we will have two different uh, values to plot, right? Okay, so that's fine. What we will do is that we will try to group this particular set of data into different clusters. So how we will do that? Firstly, we have to know how many number of clusters we want. So here we already have kind of a WCSS.py Python script where we basically, what we do is that we load up the data point. Okay. So if you remember, we when we showed the data point, we talked about that we only are interested in the column 2 and column 3 of the data set. So that's represented by this Y1 and Z1 here. And we, for calculation's sake, for quickness sake, we did not consider all of the data points, but we skipped to every 10th kind of position, and we took the data points from there. So we take a much lesser number of data points than what was present in the data set, but that will speed up the calculations by a lot. And what we do is that we use in this particular case. So if you remember, if you notice on the upper hand where the cursor is kind of highlighting, so here we have from this particular tool of scikit-learn or sklearn. Okay, so here we have imported the function k-means. So k-means tool that will perform basically our calculations, and that is what we are indicating here. So from range in 1 to 12, so basically the number of clusters ranging from 1 to 12, we will plot the within cluster sum of squares calculated value using this type of different parameters. So here you see the initialization algorithms k-means plus plus, the maximum number of iterations allowed is 300, so on and so forth, right? So just for quickly plotting, we'll just try to run this Python script, and we'll see that it has given us a .png file as per the plot, and this is the plot that is kind of coming. Okay, so here you will see on the y-axis you have the WCSS value, on the x-axis you have the number of clusters, and it seems that after, let's say, we can have cluster 3 here, number of clusters as 3 here, because after the number of clusters is increasing more than 3, so we do not see any significant reduction in the WCSS value here. So let's take the number of clusters as 3, and that's the plot our data point based on that. Okay. So for that again, we are considering the column 2 and column 3 as indicated by the Y1 and Z1. Here in this particular case, we are indicating how many number of clusters we want. For example, here number of clusters is given as 3. The algorithms, etc., they are staying as same. The number of iteration is given. What color we want to indicate our different clusters as, that is again given here. So this type of example script you will also find in the documentation. You can construct this kind of scripts yourself by looking at the documentation of how the particular scikit-learn module is working and how the particular k-means tool within that module is working. Okay, how to write the code for that. But basically, if we just run this one, we will see that a particular image is coming along with this information. Okay, so if you open up this image, you will see that the algorithm is able to divide the entire data set into these three representative clusters. Okay, the cyan one, the orange one, and the green one. And with the blue dot, the cluster representative. Representative means the cluster centroid in this case. Okay, the centroids are represented there. And on the terminal, it is pasted which centroid, basically which position centroid they are taking how much volume or how much percentage of the total data set. So in this particular case, you will see that this cluster 2 that has 4613 number of data points, that is covering overall 57.66%. So very high percent of the entire data set. And what is cluster 2? Cluster 2 is basically this minus 2.023 and 0.275, that particular point, right? So minus 2.023 is I guess coming around here, and then 0.275 should be around here, right? So that means this orange cluster, that is our kind of the most dominant group in this particular data set, right? The second most dominant group is cluster 0 here as indicated, with 2085 number of data points, 26.06% coverage of the total data. That is minus 1.217 and then 1.1014. So that means I guess this should be this one, right? The cyan colored cluster here. Okay. So that's one way to group your data. So that is called k-means clustering. And in this particular case, what we took, if you remember, for this plot, I have indicated DPC1 and DPC2. So this was just briefly, this was based on the dihedral principal component analysis of a molecular dynamic simulation trajectory. So what this kind of analysis does, it basically can be, let's say, representative calculation of the structural dynamics of a particular region of a protein where there is much higher variation in the dihedral angles, phi side angles particularly. Okay, so those regions are mainly the loop regions, unstructured regions, because they tend to fluctuate a lot, right? So perhaps from calculating this sort of dimension reduction from their dihedral properties or dihedral values, we can get to know about which conformations these particular loop regions are attaining. Okay, so that's kind of the basic of this dihedral principal component analysis. And since in any sort of principal component analysis, you will see that the principal components are modes, basically which capture the variance within the data. So how they are ordered is that the mode 1 or the principal component 1, so that will capture the maximum amount of variance in the data. And as you go down from mode 1 to mode 2, mode 3, mode 4, and so on and so forth, the significance of capturing the variance will gradually decrease. So that basically means that the first 2, or let's say the first 3 in some cases, those modes will be better equipped to give you the variation or the significant, let's say, differences or dynamics within the protein. So that's why we have used DPC1, DPC2, which are basically the first and second modes. Okay. The particular data that I showed, you can also represent it in terms of free energy plot. So I guess you can just take a look at it in this way, for example. So rather than dividing the data points into 3 different clusters, here what we have done is we have previously from, let's say, like we have apart from, or rather than taking only 3 clusters, we have taken a total of multiple, let's say 100 or 200 or something like that, same number of this kind of number of beans. So what beans usually mean is that they are a short range or short interval range where you can put each data point into, and this is based on a kind of a Boltzmann population distribution. So where you will basically see that, okay, so particular this particular bean is having this many number of data points, and if there is a bean that is having the maximum number of data points, so that will indicate the lowest energy state, because again the lowest energy state will be most stable. That means a lot of snapshots or frames will be attaining that particular state, right? So based on that kind of logic, and based on the population difference between the most populated state, most populated bean, uh, and the other beans. So based on their differences in population, we can assign each particular bean a particular energy value. So that is the sort of free energy surface uh, structure and how we calculate it. So here again, you will see that the most dominant conformation is coming from this particular range. So you can again cluster, you can again divide the data point in this particular way also by assigning the energy. In the case of molecular dynamics, that is quite often done, because rather than just taking three clusters, this will give you a kind of a more nuanced idea about where your particular um, stable structure or minima structure are forming, right, based on which values. So that was just one brief example. Mm, yeah. So just from here and then here from the other screen. That was k-means clustering briefly. Now another command or another program that Professor Gomiha also went over was called the blast class, right? So in this case, you see, um, in the case of the blast class also, you have to, of course, give it a certain command, right? For example, here it is saying -i in file, -o out file. You will see that for most programs, that is kind of the way they take the input and designate the output. But apart from that, there are other parameters also. For example, -p, then capital T, then -l, capital L, it says 0.8, then -b, capital T, -s85, so on and so forth. Okay, so here something called this -l as 0.8. What it will indicate, you can find out in this particular table or the notes that are given here. So -L will basically indicate the threshold or minimum length coverage. Okay, so if it is having a value of 0.8, so that means that the coverage for the blast class input will be, the coverage for the calculation of blast class, it will be 80%. Okay, so just since these kind of algorithms are discussed by Professor Gomiha, just try to remember the key parameter value and how they are indicated. For example, in the earlier case for CDV, we see that -n is indicating the word states, right? The word count. And then the -c was indicating the similarity. In the case of blast class, -l is indicating the coverage, right? So these kind of information just try to keep in your mind. Okay. Yeah. So again, another set of concepts that was discussed by professor was the uh, set of distances, for example, that can say how different or how similar particular amino acid sequences are. Okay, so we also saw in the previous cases also. So distance calculation is very important, right? The distances can say whether the two sequences are similar or dissimilar, so on and so forth, right? Based on certain threshold values, right? So one concept that Professor Gumha discussed was called the Hamming distance, right? Now, what are Hamming distances? How they are calculated? So basically, Hamming distance means for each type of amino acid that you have to take the composition of that particular type of amino acid or that particular amino acid for sequence 1. You have to take that for sequence 2. Then you have to subtract this value, right? And then take an absolute value of that. That is why the modulus is there. And then for all of the amino acids that are present, what you have to do is that you have to sum up the absolute value of the differences. Okay, for each of the amino acid present here. So that's how you will calculate the Hamming distance. Again, it will tell you something about the differences for these two sequences, in this case based on the amino acid composition. So it will take compositional difference, it will tell. So not positional differences for amino acid conservation, so on and so forth, but it will tell you about compositional differences. Okay, so can anybody calculate what could be the value for the Hamming distance in this particular case? Just take each of the amino acid that is present, that could be present here, uh, calculate the composition for sequence 1, calculate the composition for sequence 2 for that particular amino acid, subtract composition in sequence 1 minus composition in sequence 2, right? You can do the vice versa also, because ultimately we are taking an absolute value of that, and then you have to sum up for all of the amino acids, okay, using this particular formula. So just try to calculate it out. We'll just look at it briefly. Okay. So I see one option B. Yeah. So how do you calculate the composition? Of course, you first count down how many number of that particular amino acid you have, and then you divide by the total number of amino acids presented. For example, um, for alanine, right? So in sequence 1, how many would be there? So 1, 2, 3. Then you divide that by the total number of amino acid sequences. So I see it should be 10 here for simplicity's sake, right? Then you will get the composition of 0.3, right? 3 divided by 10, 0.3. Again, for the next sequence, or the sequence 2, you will have to calculate the same thing. So the composition of alanine in this particular sequence. So in this case, what you will see that 1, 2, 3 alanines are present here. So if you calculate the composition of for sequence 2 also, you will see that you will get a value of 0.3. In fact, for all of the amino acids that are mentioned here, okay, so this should cover all of them. You will see that both in sequence 1 and in sequence 2, they have same composition values, 0.3, 0.3 for alanine. Let's say for aspartate, you have 0.1, 0.1, right? For isoleucine, 0.2, 0.2, so on and so forth, right? So this will cover the entire stage of amino acids that are present here, and all of them have same composition across both of the sequences. So that means that for each value that you will have to subtract here, for each subtraction calculation, you will get a value of 0. If you sum all of them together, you will still get a value of 0, right? So ultimately, you will have the Hamming distance of 0. That means that compositionally, these two amino acid sequences are quite similar, okay, because their compositions are same for particular amino acids that are present here. So that is how you can calculate the Hamming distance. Okay, basically, yeah, you do the subtraction for composition in each amino acid's case, you will see a total value of 0. Okay. Yeah, so that was Hamming distance. Let's say we do one more problem for today. So here it was, the question was asking is that you have to calculate the sequence identity between two given sequences. Okay. Now, how do you calculate the sequence identity? Uh, Basically, if you have a different number of components in each of the sequences, you have to do it via this formula. So number of identical residues via the length of the shorter sequence. Just remember that. Okay. So what will be the identity value here? So identical residues will basically mean the exact same characters, and then the shorter sequence, that's length we have to divide it by. Okay. So if you see the calculation very quickly, yeah, 80%. Exactly. So number of identical residues will be 8, and the total number of amino acids are 10 for the shorter sequence, right? In this particular case, I guess they are the same value, but if there was different kind of length, you have to calculate for the for the shorter, you have to use the shorter sequences length. Okay, so in this case, it would have been 80%. Okay, so these types of questions, very simple numeric based questions, could come in your assignments. Just remember the type of formulas that Professor Kumiha mentioned, for example, in Hamming distance, so on and so forth. Okay, so those you could again encounter in your assignments. Okay, okay. So for today's class, I think I will just wrap up here. Yeah. So yeah, for today's session, we'll wrap up here, and then for the next week's content, again, we'll meet on the next upcoming Monday. There will be another tutorial session that is going to occur tomorrow, Tuesday night, in the exact same time, I guess 6 to 8. So try to attend that one also. Try to go through Professor Kumiha's lectures very carefully. Yeah, and then for next week's content, we'll meet on the upcoming Monday. Okay. So thank you all for joining the tutorial session today. And yeah, we look forward to the next week's lectures and contents. Okay. Thank you so much.